Okay, so good afternoon, everybody, or good morning. I'm very happy to be here today, and actually I have to say that I'm also happy to present while I'm sitting down. Otherwise, I think that my legs will be shaken if I would be in a normal conference. But yeah, I'm super happy that I can virtually see many people from the INA community. The first thing that I want to say is that I want to thank the NannoTalks committee, and especially Baptiste, Clara, but also Felipe for inviting me and for check that everything was fine. And yeah, I'm a postdoc at the University of Milan. I think that most of you knows me. And today we're going to talk about the response of coccolithophores, mainly in terms of growth to trace metals released from volcanic ashes. That one is one of my favorite topics, so I'm very excited to talk about that. The second thing that I want to say is that, of course, this is not my a project that I'm doing by myself. So I want to introduce my co-author that are coming from different institutes. So first of all, from the University of Milan, I collaborated with Elisabetta Erba. I think that everyone knows her here, so I don't have to introduce her so much. And I also collaborated with Chiara Bettoni, that she's now a PhD student of our department. I'm going to talk about experiment that I performed in Germany at the AVI in Bremenhaven, and I was part of the research group led by Scarlett Rimborn, with whom I discussed a lot about how to perform these experiments. And I collaborated with Florian Koch, and also I really needed the help of Christian Volkner. And then I also collaborated with Katrin Wuttig, that she's one of the most enthusiastic scientists I've ever met. She's a chemist, and she was working at EMAS in Hobart at that time. And finally, the last person of this big group of people is Lynn Hoffman. I have to say that this project is part of her pet project that she has always wanted to do. So I'm very, very happy that I was able to work with some of her volcanic ashes. So now it's time to talk about science. And another thing that I want to tell you before talking about the experiment is from where we took inspiration from. So I'm going to take a while, but I think it's quite important so that you understand why we decided to do this experiment and what we expected from them. Of course, we took inspiration from the geological record. This is an example. Actually, it's not even an interval of time that I've studied, so I'm not an expert here, but of course, around the Eocene-Oligocene boundary, there was a big change in the climate. It is considered one of the most prominent global climate reorganization of the Cenozoic. It's a time where the Antarctic ice sheets appeared and where the temperature decreased a lot, as suggested, as evidenced by the oxygen isotope. And well, there are many hypotheses that try to explain this variation in the climate, this turning point from a greenhouse climate to an icehouse climate. I'm not going into that too much. But what I liked a lot was when I read this paper by Jica et al. In 2009, in where they estimated the number of volcanic eruption around 35, 34, 28 million years ago, and they counted more than 500 eruptions. That means that there was one eruption every 13,000 years. Some of them, they were closer, some of them not so close. And so they asked themselves if this volcanic eruption could have influenced the climate. These volcanic eruptions were subaerial, and so there might have been different consequences. For example, these eruptions produced for sure gases like sulfur that, for example, sulfur generally react with and produce sulfuric acid that can be backscattered or reduce the solar radiation that reach the surface of the Earth. So this might be one reason for which we have this reduction in temperature. But another possibility is that this very extreme, very large eruption could have fertilized the surface of the ocean because they produce ashes, and these ashes could have promoted the algal growth that in turn would have reduced the carbon dioxide in the ocean and in the atmosphere. So in this paper, the author they hypothesize that volcanism could have been a kick for additional cooling and reduction in carbon dioxide. If we go not so far away from today and we look at this reconstruction made by Carter et al. In 2009, and we look at the carbon dioxide anomalies, you can easily see that after the very big eruptions, like after the Mount St. Helens eruption or the super famous Pinatubo eruption in 1991, there's a reduction in carbon dioxide. It is easy to think that one possibility is again that the ashes produced by this eruption reduce the radiation, the solar radiation, and so they reduce the temperature of the Earth for some years. But it's also easy to follow this equation. Volcanic ash supply trace metals on the surface of the ocean. Trace metals fertilize the ocean. They increase the photosynthetic conversion of carbon dioxide to organic carbon, and in turn, they decrease the oceanic carbon dioxide. Let's say in a very easy way, it's like if the organic carbon pump was accelerated, and therefore we can imagine this because the settling and the boreal of fraction of particulate organic carbon could have induced a long-term reduction of atmospheric carbon dioxide and global cooling. But as I think that most of you know, in my research group, we study for quite a long time, and we're still studying the Cretaceous oceanic anoxic events. And this is just to tell you where we took inspiration from. And during these oceanic anoxic events, we studied and we observed variations in sizes and morphology of calcareous nanoplankton. With sizes, I mean that we saw a reduction of some species that we call dwarfism, and we also observed some malformation, meaning that some species modified their shape, they became more or less elliptical. And we found out that these variations in the oceanic anoxic event, for example, in the oceanic anoxic event 1A of the Aptian and oceanic anoxic event 2 of the Cenomanian Turonian, were coeval with trace metal peaks. In that case, we are talking about a different kind of volcanism that produces trace metal peaks, because Cretaceous oceanic anoxic events are related to the formations of large igneous provinces, therefore to submarine volcanism. But the release of these metals from this volcanism at the bottom of the sea, these trace metals possibly have enough buoyancy to reach the surface of the ocean, and they might have influenced the phytoplankton community. So when we took all this information together, we decided to have a look at what could happen or what is happening nowadays in living coccolithophores, and we decided to perform some experiments. Now I'm going to tell you something about this experiment. We performed experiment using 3 different volcanic ashes. Why we decided to choose these 3 ashes? One is the ash from Eyjafjallajökull from the eruption in 2010. One is from the Greenstone ash, again from Iceland, from the eruption in 2011. And the last one is from Chile, from the big eruption of 2015 of the Calbuco volcano. We chose these 3 ashes because we needed quite a big amount of ash to perform this experiment. And actually, it's not so easy to get ashes after an eruption, because to perform this experiment, we needed ashes that didn't take any rain. So someone needed to go and get them as soon as possible. So they're like diamonds for me. Because they're super precious. And we performed this experiment, as I said before, at AVI. And we use a trace metal clean technique. So we use a procedure that allow us to reduce the contamination from other trace metal. That is quite challenging and took quite a long time. And for that reason, the experiment were performed in duplicates, because it was, it was a very long procedure to have all the material that we needed clean and ready for the experiments. Another big thing of this experiment is that we use as a background water for our experiment, the water collected from the Southern Ocean and from the Drake Passage. This was a big gift from the AVI. And so to prepare the water for our experiment, we added to this natural seawater 3 grams of ash for every liter. And then the procedure, we had to shaken these bottles with ash and water for 10 minutes. Then we left everything to be settling, and then we filter this water plus ash. What we did subsequently was to put this solution of water plus trace metal released by the ash with different concentration. And at the end, we obtained 7 different concentrations of ash that goes from 0.15 micrograms up to 3 grams of ash per liter. Why we decided to do that? Because we wanted to imagine to be next to the source, and then more ash, we expected more ash to be settled. And then we went from the source to far away from the volcano. And so we imagine a lower amount of ash inside our bottle for our experiments. What we used for our experiment, where we wanted to be consistent, and so we used 2 strains of EX, of Emiliania axillei, that were also coming from the Southern Ocean. So we took these 2 strains, 503 and 530, that we took from the Australian collection. And just a few more notes about the experiment. In our experiment, we didn't put any trace metals or any chelate like EDTA, because since we were using natural seawater, we wanted to see how the algae reacted using natural chelate. There was one exception, because in our experiment, we used 2 controls, let's call them like that. In one control, we didn't put anything, so just natural seawater, no chelate, no EDTA, and no trace metal. On the other side, so on the opposite side, we put trace metal following the receipt that is called F-2 and EDTA, so also the chelate. That means that in that situation, the algae had everything they needed, all the metals that they needed to grow. And in between, there are the ashes, different ashes concentration. The experiment, the experiment ended when the algae were still in their exponential phases. And another thing that I want to say is that we were very strict in the way we took samples. So we took sample every 2 days at the same time of the day, because the life cycle of the cell changed through the day, and so we wanted to compare the data, and so we were very strict in the timing of our experiment. What we look at the cell concentration to calculate the growth rate, at the cocosphere and the cell size, and also at the photosynthetic efficiency, which is a measurement of the chlorophyll fluorescence, and this is fundamental to understand the performance of the photosynthetic apparatus. It means that when the values are low, the algae are under any kind of stress, maybe nutrients or trace metal limitation. When the values are high, the algae are able to photosynthesize properly and are not under any kind of stressful conditions. Then, of course, we look at the coccolith produced by these algae. So we made some measurements about the size of the coccolith, the number of the different elements that compose the coccolith. And we look also at we quantify the shape of the coccolith. So we quantify the number of malformed, incomplete, or normal coccolith as well, of course. Okay, it's time to show you the results now. And the first result is that we don't have result for the Chile ash experiments because our algae didn't grow at all with this ash. We were very disappointed because actually this was the first experiment that we performed. And so I asked myself many times if I did something wrong. And so we performed this experiment twice and we obtained the same result. I will try to explain you later on. We try an explanation for this, and I will try to explain that later, but just keep that in mind. So I can show you only the result for the 2 Icelandic ash, and I will guide you through this graph now. This graph represents the growth rate, so how fast our algae grow under the 2 Icelandic ash. On the left side, the graph represents the strain 503, and on the right side is the strain 530. The gray columns represent the 2 controls, so the natural seawater on the left side of each graph and the other 2 on the right side of each graph. And then the white columns are the experiment with the Grimsvötn volcano, and the light blue column is the growth rate of the experiment where we put the Eyjafjallajökull ash. The first thing that I want to say, and I think it's easy to see, is that the growth rates are generally quite low, even under other 2 conditions. I was used in the past to do experiments using artificial seawater and actually using strains coming from the northern hemisphere. So I usually had like double the growth rate that I observed in this experiment. So I have to admit that I didn't sleep many nights thinking about this growth rate data. But at the end, luckily, we found this work by Muller and co-author 2015. In which he studied many strains coming from the Southern Ocean of EX. And even under optimal conditions in terms of lights and temperature, even higher temperature and carbonic chemistry, nutrients, trace metal, and so on, the growth rate was always smaller than 0.5. So looking at our data, okay, finally we figured out that we were on a safe side because under other 2, the growth rate was similar to what Muller et al. In 2015 found. Then you can easily see that the 2 strains behave in different ways to ash addition. The strain on the left side, 503, increased its growth rate when we put the Grimsvötn ash significantly. While the growth rate didn't increase, it was very similar to the natural seawater growth rate when we added the Eyjafjallajökull. For the other strain on the right side, so 530, is just the opposite. So when we added the Eyjafjallajökull ash, this strain grows nicely and faster, while its growth rate decreased when we added the Grimsvötn ash. Then we look at the FEFM. As I said, this is the photosynthetic efficiency of the algae. And the data are really nice. So if you look at the left graph for the strain 503, you can see that in natural seawater, the FEFM value is super low. It means that the algae were under some kind of stress, possibly due to trace metal limitation. And for both ashes, the FEFM values increase. Let's say that FEFM values are good when they are above 0.35, let's say. So the algae were in a good shape when we added ash in that case. For the other strain on the right side, so for the strain 530, the FEFM values is not so bad in natural seawater, even if it's not optimal. And it increased a lot under the Eyjafjallajökull ash addition, while for the Grimsvötn, the FEFM values are very low, and it follows actually the growth rate pattern, if you remember the slide before. Then we look at the coccolith size, and I have to admit that nothing much changed. The only thing that we observe is that under F02, so when the algae had all the metals they wanted and all the chelate they wanted to that make the metals bioavailable, the coccoliths are smaller in size. Actually, we didn't dig too much into that. We just asked ourselves if there was a relationship between the coccolith size and the growth rate, because under FOTR2, the growth rate was much faster and the coccolith are smaller, but it's not really the topic of this work. So I'll just leave you with this comment. But what really interested us a lot was that we found out that when we added the ash inside our water, both strains produce nicer coccoliths. So there were less malformed and incomplete coccolith. Or maybe I can say that in the other way around. So without ashes in natural seawater, the percentage of malformed, incomplete, and malformed coccoliths, malformed and incomplete coccolith were super high. If you look at the graph on the right side for the strain 530, the percentage of normal coccolith are even lower than 20%. And so it seemed that there's a positive effect of ash on Emiliania axle calcification for these two strains. Well, now after I show you this data, I will spend some time in front of this table because this is what guide us in our interpretation. These are the analyses that we perform at the ICP-MS to see the amount of trace matter released by our ashes. And what we found out, and they are expressed in nanomolar. And what we found out is that, first of all, for the natural seawater, so the background water, actually there were very low concentration of some trace metals like manganese, iron, but also zinc is not very high. And this can explain the reason for which EX didn't grow faster with natural seawater, where this water is coming from the Southern Ocean, so we expected this kind of values. For Eyjafjallajökull and Grímsvötn, actually this trace metal concentration increased, especially manganese and iron, but also zinc So we think that the increase in growth rate that we observe in some strains in some cases could be due to a fertilizing effect of these two ashes. Then when we looked at the Chile ash, we were super surprised of this value, of the copper value that is extremely high. And we straight away thought that it could have been toxic for the growth of our algae. And so this might be the reason for which our algae wasn't able to grow with this ash addition. Actually, we found out in this paper by Levy and co-author 2007, they calculated that with a value above 15 microgram per liter of copper per liter, EX wasn't able to grow and to reproduce anymore. And actually, if you transform nanomolar, 172 nanomolar of copper correspond to 17 microgram of copper per liter. So it makes sense, it might be a possibility. But I want to stress out that the picture is not so easy because it's not only a matter of concentration, but it's also a matter of ratio between different elements. For example, there is a possibility that that the manganese was the one that induced the that didn't protect, the algae, because the manganese, it is known, at least for diatoms, that the manganese can block the uptake of some toxic metal into the cells. And if you look at our data, it's easy to see that in the Chile ash, the manganese is very low compared to the copper, while in Eyjafjallajökull it is, and Grímsvötn is much higher if you make a very easy ratio between these two elements. Furthermore, this is an artificial experiment. So It is also possible that the algae were also stressed in any other way. And so, for example, the cells could be struggling for many other reasons. And so what we've seen for the Chile ash could be related, for example, to too low trace metal concentration of some other elements. What we know about the literature, so if we look at papers that were already published, well, Lynn Hoffman actually, she performed an experiment in 2012 using two different groups, so diatoms and coccolithophores. So she used Thalassiflora pseudonana and Emiliania axley, but she used a strain coming from the North Hemisphere, and she used natural coastal seawater, I think from the Baltic or from the North Sea, I'm not sure. And she tested different volcanic ashes from the Pacific Ring of Fire. If we look at Emiliania axley results of this experiment, so this is the growth rate, and what she found out is it's that even if they increase the concentration of the ash from 0 up to 2.67 gram per liter, in most cases, the growth rate remained very stable. Why? One explanation is that the background water that they used for this experiment, so the coastal natural seawater, was already very rich in trace elements. So if you see the data down in the table, you see that iron, manganese, nickel, and zinc were already quite high. So the only situation where Emiliania axley changed its growth rate is when they tested the Sakurajima ash. This ash actually recorded the highest trace metal concentration. So it is possible that this ash was actually too toxic for this algae. In 2017, we also performed another experiment. We used different trace metals. We tested different species, but we used, first of all, artificial seawater. We added trace metals, we added EDTA, and then we put a big amount of 4 different elements that were zinc, nickel, lead, and vanadium. And in that experiment for EX, we observed actually a reduction in the growth rate when we added these trace metals. We observed reduction in the coccolith size, and we also observed an increase in malformation. So it's just the opposite of what I told you about our experiment until now. So to summarize all this data, we produce this graph. Is very, very easy. Of course, it doesn't tell the whole story, but we think that this might be what could happen in a natural situation. So when you are in a coastal area, the trace metals are already enough. So we are under replete condition. And so if you put more ash, so if you imagine a volcanic eruption, it is possible that this volcanic eruption do not stimulate EX growth. But if you are in an open ocean or in an area like the Southern Ocean that is characterized by low trace metal concentration, it is possible that some volcanic ashes could stimulate and fertilize the ocean, producing and that this can increase the growth rate of EX. And finally, if you are in a coastal area and you increase the amount of trace metal because there's a big eruption that has a very high concentration of trace elements, you are on the toxic side of the story. And so it is possible there that the algae are not able to properly grow and they strongly reduce their growth rate. Just one last comment. These are experiments performed in a lab, but they perform experiment in natural condition as well, or observation. For example, for Eyjafjallajökull, actually for the ash that we use, so after the eruption in 2010, Akteber and co-author, they found out that there was a chlorophyll peak in May. Generally in that area, there's a seasonal bloom that occurs later on in July or in June. And so this bloom was before, possibly because of the fertilization of the ash released by this Eyjafjallajökull eruption. And in another experiment, was an experiment performed by Browing et al. In 2014 in the Drake Passage and North Scotia Ridge, they observed that when they added volcanic ash and volcanic ash plus iron, there was an increase, as you can see in the graph at the bottom, an increase in the FeFM values, meaning that there was an intense photosynthesis, and so the algae were fast, they reproduce faster, and they like these conditions. However, in this experiment, what we don't know is which species actually benefited by this ash, in the increase in the trace matter released by the ash, because they didn't look at the species composition, and they mainly know that the phytoplanktonic community benefited by the ash release. So these are my take-home messages at the end of the talk. So we observed in our experiment that we have different responses of the two strains tested from the two Icelandic ashes. And so we underline that there is a difference not only between species, but also between strains. Something I didn't say before is that in natural communities, the responses of the phytoplankton could differ due also to the presence of organic ligands that you can only find in natural conditions, because they are produced by bacteria or by some other phytoplanktonic group. And the influence of trace metal depends on not only on their concentration, but also on the ratio between different elements. And so we hypothesize that in natural environments, volcanic eruption will likely affect species composition and even strain composition within species. The last thing I want to say is our open question. I mean, we are very positive and we love Emiliania, so this is a positive, let's say, question. We ask ourselves, does the response that we found in our experiment give EX an advantage compared to other species? Because there are many different strains, and so it is possible that under any kind of volcanic eruption, at least one strain in a natural environment might survive under toxic condition. And I thank you for being here and listening to me.
Thank you, Julia, for a really fantastic talk. The forum is now open for questions, so you can either raise your hand if you have a question and you'd like to ask it directly, or you can type it in the chat if you'd like to ask your question via me. Okay, so I see Alex has his hand raised. Alex, do you want to go first?
Actually, it was just that I was saying that was a really nice talk. I thought that was really interesting.
Thank you.
So I hadn't realized about the manganese helping with trace metal uptake and also inhibiting the toxic effects. I thought it was quite interesting.
Yeah, it is known for diatoms, but they are not really good data for coccolithophores, and it's always like that. But for diatoms, it is like that. I mean, the more manganese, I mean, there's a threshold values above which manganese is toxic for diatoms as well. But below this value, it helps a lot in, yeah, in not taking too much toxic elements inside the cell. Mm-hmm.
Excellent. That was kind of just a comment.
Thank you.
Thanks. Next, Mario has his hand raised.
Yes. Well, I have to say that I was really looking forward for hearing someone talk about this subject, you know, and I have so many questions that I will not put every single one, because I would dominate probably the meeting, and that's not fair for the others. But we have to talk. Probably my proposal is that we talk eventually later on
whenever you want.
Some other, on some other, but for now, I start to look for this perspective from a completely different perspective. Was when I started to study coccolithophores around volcanic islands. I start to wonder if the volcanic activity would be beneficial or could be somehow
detrimental,
harmful to the development. Because in some cases I see a good fossil record or present day assemblages, and in other cases I don't. So probably it plays, as you said, in both ways. It can favor some, it can restrain others. You performed the cultures. That was something that I would love to do, and we were limited here. I was very limited to do it, and I couldn't do it, but you performed those, so that's great, because that will give complementary data to some of our perspectives. But for instance, I would select, I don't know how you, well, you said you selected the volcanic ashes because they were more available. You had the opportunity to have them. And by the way, my congratulations to say the Icelandic name, because I can never say it. It's always a struggle to say the name. So congratulations on that. So my approach would be to consider, for instance, a mafic ash and a felsic ash. I don't know the nature, the volcanic nature of these ashes. If they get in your last slide, the authors had that concern, I think, because I saw there mafic or felsic or basaltic and rhyolitic.
Well, we were more interested in the trace metal released by these ashes, and also this depends on the water that you're using. So it's quite a complicated task, and unluckily for this experiment, we could use ICP-MS only at the end of the experiment. So we went blind somehow, because we didn't know what was inside our ashes, because the ICP-MS was broken as usual. And so, I mean, I think it's, I mean, there's so much to do with volcanic ashes, because volcanism is something that happens sometimes and in so many different ways. So there are many different variables here. So as I said in the last graph, it's not, this was a very easy way to represent our result, but there are so much more things to think about, because as I said, this is also an artificial kind of experiment. So for sure, we can also look at different kind of volcanoes, thinking about different sources. But as I said, it's not even so easy to find good ashes to use in the labs, and the amount that, enough amount of ashes, I have some of them here, but they are not enough for performing this experiment. So of course, makes sense to try different kind of volcanoes with different composition, for sure.
Well, you showed that it's important because you used several, and some had a completely different. Well, what we did was to look for the assemblages on cores immediately before and immediately after an ash event. Okay, so in situ, on the bottom sediment, and we could see that the behavior of the assemblages changed from one ash layer to the other. There was not always the same behavior. Some species increased, some species decreased, but it was not always the same behavior. And the volcanic ashes were different, had a different composition. I didn't know the trace elements, but they were more mafic or more felsic. So they had to play, for sure, different role. Well, we haven't published that data yet. And now I have 2 additional cores where we have also just before and just after volcanic ash layers to test again, to see what happens to these assemblages. But I would like very much, I would not take much more time, but I would like very much to discuss these results with you probably later on and see how we can combine our data, our approaches to see if our data can be also meaningful for this question of the influence of volcanism in general for the coccolithophores. So
My
thank you very
pleasure.
much.
Definitely. Thank you.
Okay, are there any other questions in the room? Otherwise, I have one in the meantime, if nobody has their hand raised. It was a super interesting talk, and I was interested in whether the addition of more and more ash in your experiments had any effect on the in situ light intensities, and therefore that might have affected the growth rates.
Oh, light intensity, not at all in our experiment, because of course we filter our water, so we didn't have variations in the light. Yeah, this is also another issue, because of course, when you have a volcanic eruption, ashes could be on the surface of the water, and they definitely can influence the euphotic zone in that sense, for sure. But of course, not in this kind of experiment. Yeah. So
do you think that if you actually directly added the ash to the water without filtering it, then all the cells would not survive? Or do you think
I don't know. I don't know. It was something else that we thought about, but at the same time, I don't think it's exactly the right procedure, because then, I mean, it's much more easy that the algae settle at the bottom of the
you're mixing
bottle, and then you have to turn the bottle every some hours. So I think in an artificial experiment, in a lab experiment, I mean, it's better like that. I would say so.
Thanks.
You're welcome.
Well, if nobody else has questions, I can. I know that, sorry, can I go? Okay. I understand the importance of the trace metals, but have you also checked the other elements, the other more common elements? I mean, the calcium, the silica, that in, well, probably not with the millennia, but in some coccolithophores we know it also plays a role. So what about the main elements? Did you control that as well?
No, since it's natural seawater, everything was already fine for them. We just put as usual nutrients and vitamins, because this is how you do for these kind of experiments, but we didn't modify anything else. We just check the pH, because of course, if you put some ashes, the pH could have been changed, but it didn't. So, and we didn't do anything else. I mean, they definitely had anything else they needed. If you see the FODR2 conditions, so where we put trace metals as well and chelate, they grew nicely. So I think it's not a matter of calcium or anything else. Yeah.
Okay, I have a question on the chat. Very nice talk, Julia. Congratulations. Did you find any correlation between the amount of trace metals and coccolith malformation?
You mean which kind of trace metals and malformations, or? I mean,
between the concentration of trace metals and malformations, I guess, but I guess this question comes from YouTube, so I'm not sure.
Okay, so what we found out is that when we put ashes, the malformation decrease, that's for sure. But it's really, really hard to say which could have been the elements that could reduce the malformation or so on, because different hashes has different elements. So it's not like that they are so similar and that you can see this one or the other one. We tried to find out in literature if there were some data about calcification and trace metal, but we couldn't find anything. So if the question is, do you know which trace elements could reduce the malformation? Actually, I don't know. The only thing that I can say is that with volcanic ash, the malformation are definitely reduced. Or, as I said in the other way around during the presentation, without volcanic ash and trace metal, the malformation and incomplete coccoliths are with super high concentrations. And I prefer to say like that because I don't know which elements could have reduced this malformation and so on. Yeah,
it's really interesting to think of the default state of the coccoliths being malformed, and then when you add in excess these things, then they become more and more perfectly formed.
Yeah, but you can really see it from at the SEM. I mean, I saw a few filters just after the experiments, and it's there. Just when you put the sample, you easily see these variations in morphology in natural seawater, and when you add trace metals or ash, it's very, very evident.
Okay, so I have another question that's coming on the chat from Jorge. Can you please ask Julia to talk about those molecules that exist in seawater and allow the intake of trace metals by coccolithophores?
Sorry, I don't understand. I mean, I don't know if I understand the question or if I can.
The question is related to how do the coccolithophores take up the trace metals?
Oh, I mean, there are many channels that allow them to take the trace metal inside the cell, but I don't know which elements can get inside the cell because there are different different ways, and I'm not a physiologist, so actually I want to stop myself here before saying silly stuff. So yeah, I think I cannot reply any further.
Okay. Baptiste also has a question. Thinking back to Gerald's talk last month, where it was revealed that silicon is very important for coccolith production, could it also be the case that this element is essential for coccolith crystal formation?
Yes, for sure. I mean, I collaborated in a paper by Cinzia Bottini that some of you might have seen. She performed some analysis and she found out that some trace elements were incorporated inside the coccoliths. I mean, she used some of the samples of the experiment I performed in 2017. And she found out that some elements were incorporated into the coccolith, while some others were just on the surface of the coccolith, so they might not have been incorporated. So it's a different task because it's not so easy to have this kind of analysis, but yeah, it is possible that they play a major role in coccolithophore calcification. Not all of them for sure. I think that sometimes it's more a matter of, let's say, stressful conditions. So I think that the coccolithophores are not able to calcify properly because something is missing. They are not really happy, and so they just produce ugly coccosphere, like making it that way. So I'm not sure that it's always a matter of the coccolithogenesis process, but it's more a matter of more in general the physiology of the cell. And yeah.
Okay, thank you. I think that's all of the questions that have been directed at me. I don't think, apart from Mario, is your hand still raised because you have another question?
Now, Juliana,
wait, Juliana has a question. We'll let you go first.
No, mine is not a question. I just want to thank, if anybody else has another question, but anyway, I just say that I want to thank very much Julia for this interesting talk and excellent project that we really needed something new like this, very interesting. And I want to thank Clara and all the other guys, Baptiste, Felipe, and the Aurora team for this producing this NannoTalks. They keep in touch our community. Thanks again.
Thank you.
Thank you all for attending.
Juliana, your filter is very good. It looks like you're in the field.
It's quite windy.
It's a very good filter, very good Zoom filter.
Yeah.
Yeah, a nice day, nice windy day.
Okay, does anyone else have any comments on for
Just
Julia?
one.
Jeremy.
I was a bit this thing about having lots of malformation in the control sample is a little worrying, because if you look at plankton samples, normally the malformation rate is really low.
I know.
Culture is often high, so I mean it's really difficult to do this kind of experiment. I would prefer to have your inoculum growing nicely.
Yeah, it's true.
It's difficult to. I mean, obviously something was interfering with them. It's very difficult to guess what that might be. The default position out in the ocean is definitely that coccolithophore, that they have a low malformation rates.
Yeah, that's true. I know that I don't know if it was I don't know if it was Langer or someone else that performed this experiment using different bottles, and to see if you perform experiment using different bottles, if you have more or less malformation, and if you put the bottle on one side or the other side. So I know for sure that in lab experiment, the percentage of malformation are much higher, but for sure here in the different conditions that we tested, there is a strong increase in malformation with no ash and no trace metals compared to the other one. So this is something that I'm sure about, but it makes sense to think that in natural environment, this percentage could be much less. So yeah, for sure. Nice comment.
And so you didn't add any macronutrients to your...
I added, yeah, phosphate and nitrate for sure.
In the
and
natural,
vitamin.
so to what? Well, in your control samples, you did add macronutrients, but not to F2 level. To what level? To what level? F50 or?
F20, I think. Yeah.
Okay.
Yeah.
And you don't.
And that question was also asked by somebody in the chat. They asked what the concentration of phosphorus, nitrogen, and silicate was in the Southern Ocean water that you used.
Yeah. So just normal recipe, 16.4 and yeah, and F other 2 for vitamins, and for F20 for vitamins and nutrients. Yeah. Sorry. Yes, of course.
And did you test if you had any nitrate and phosphate effect from the ash? No. I guess nitrate not, but phosphate conceivably could.
Sorry.
Phosphate is more possible, of course, than nitrate.
Okay. No, but we didn't. No.
Anyway, nice talk. Thank you.
Thank you very much.
Okay, so I think we've covered all the questions that have been asked in the chat or that people have in the room. So I guess once again, thank you very much, Julia. And thanks everybody for attending, and thank you to Aurora Science Communications for helping us put these talks on. And we very much look forward to seeing you next month for another Nano Talk.